Hi guys, Miss King here, and in this screencast we're going to be looking at the process of protein synthesis. And it's actually going to be divided into two separate videos. We're going to get the first part of protein synthesis in this video, and the second part will be in a different one. Before we can look at protein synthesis, we need to take a look at the parts that are involved in protein synthesis, and those parts are called RNA. RNA stands for ribonucleic acid. Okay, and there's three types of RNA. The first type is called messenger RNA, or mRNA, and its job is to carry a copy of our DNA instructions to a ribosome. Why does it need to do that? Where's the DNA going to be located in a cell? It's always going to be in the nucleus. Okay, the DNA cannot leave the nucleus, but we build proteins on ribosomes, and ribosomes aren't located in the nucleus, they're located in the cytoplasm. So we need to get the instructions for our protein out of the nucleus and to our ribosome, and our messenger RNA is going to do just that. So we need to be able to recognize these guys in a diagram, so let's draw one. Okay. RNA, this is going to be our sugar phosphate backbone, just like with DNA. And then we're also going to have our nitrogen bases. Okay. Now, in RNA, our nitrogen bases are going to be guanine, cytosine, adenine, and uracil. Okay. U equals uracil. There is no thymine, no T's in RNA, in any of the three types of RNA. There are no T's ever. T is going to be substituted with U instead. Okay. All right, so we have bases. It's a single strand instead of a double strand. And our sugar is going to be ribose instead of deoxyribose here in our backbone. And then we also have, since this is a message, our messenger RNA is carrying instructions like a message. The words that make up our message are going to be each three bases in a row. So 3 bases in a row are called codons. Okay, so these are like the words in our message. So this is one codon, this is another codon, and so on. The second type of RNA is called ribosomal RNA, or rRNA, and its job is to simply be the ribosome. It makes up the parts of the ribosome. So if we were to draw a ribosome, it actually kind of looks like one big blob and then an empty space and then a smaller blob right below it, kind of like that. And this rRNA is all throughout both the large and the small part of the ribosome, just making up the structure. And we have this little space here in the middle because that's where our mRNA is going to bring the message. Okay. And then our third type of RNA is called transfer RNA, or tRNA. And its job is to bring our amino acids, which are the building blocks of proteins, to the ribosome. So tRNAs are usually pretty easy to recognize. They actually kind of look like a T. So we have this kind of long part and then kind of like two arms sticking out and then another arm coming down. So here's our sort of T shape. And then at the bottom, there's usually a small section for our base pairs. So we'll have 3 bases on our tRNA. So let's put 3 bases on there. Again, there's no T's in RNA, so if we see a base in the tRNA, it would have to be a U. And again, you'll notice there's 3 base pairs in a row. Because this is tRNA and not messenger RNA, this is going to be called an anti-codon. Okay, it's called a codon on the mRNA. It's an anti-codon on the tRNA. Alright, let me change my pen color one more time. The last part of our RNA is this section at the top. We'll just make a little circle for it. And that is our amino acid. Okay, so now that we're kind of familiar with the structures that we're going to be seeing for protein synthesis, let's go ahead and look at the process. All right, so protein synthesis is divided into two parts. It is a process, so we're building our protein in a step-by-step sequence of events. Transcription comes first, translation comes second, and I remember that because C in transcription, C comes before L in the alphabet. So transcription comes before translation. And you might be thinking, why in the world do we need proteins? What's the big deal? Why do we need to even know how proteins are made? Well, there are 7 reasons that we've looked at so far. The first one is for our metabolism. Enzymes make up a big part of our metabolism, and proteins are enzymes. They also make up several body structures, like your body parts. Hair, fingernails, eyes, all those different body parts are made of proteins. They're needed for movement. You wouldn't be able to move or have any kind of motion without proteins. Remember, antibodies for defense, for our immune system. Storage in animals like chickens with their eggs. Transport, remember membrane transport, active and passive transport requires the protein carriers, and molecules like hemoglobin transport the oxygen in your blood. And then also hormones. A lot of our hormones are made of protein components, and those are signals that are sent throughout our body, chemical signals. Okay, so if we were to look at some of those examples, here are some structures. When we look at our proteins, for example, this protein might say that you're going to have blue eyes. And this protein, what's different about it is we change this amino acid to a different amino acid, and so now it changes the eye color to green. Okay, same thing here. If we have proline in our amino acid chain, then we'll have brown hair like Bella's, and if we change proline to tryptophan, then we'll have blonde hair like Edward's. Okay, and then this is a earlobe that it has a nice lobe to it. It kind of hangs down. But if we change serine to asparagine, then our earlobe will be attached, and so it kind of goes away. It attaches directly to the head instead of kind of dipping down like this one does. Okay, so whatever our proteins are tells our body parts what they're going to look like and how they're going to be structured. So it's really important that we know a little bit about how they're made. All right, so exactly, life would not exist if we did not have proteins. All right, so for step 1 of protein synthesis, transcription, what we're going to be doing is copying our DNA's instructions, the genetic code, onto an mRNA molecule. This has to happen in the nucleus because that's where the DNA is located. And we're only going to be making these mRNAs where the gene is located. And there's lots of genes on our chromosomes, so whichever protein is needed, that's the gene that it's going to use for the instructions. All right, so we have several steps that we're going to go through to do our mRNA, to get our mRNA. The first step that's going to happen, number 1, is that we're going to need to start with DNA and find where our gene is going to be transcribed. So we have to locate our gene. Let's see, let's use blue as a pen color so we can see. All right, so this picture right here, this is my DNA, this little section right here, this is the DNA. Okay, we can see the double strand, then our base is in the middle. And right here would be what's called a promoter. Let me make my pen smaller. Okay, this is a promoter site. Promoter site. And basically a promoter site is just where the gene begins. My pen is not cooperating. There we go. The gene begins. Okay, so this whole section right here, this whole area, this is our gene. This is the part that we want to use to get our instructions for our mRNA. All right, now right here, this is the end of our gene. And the end of the gene is called the termination site. Termination site. Okay. All right, so we found our gene. We know what we're going to be transcribing. The second step is going to be to unzip and unwind our DNA. Now, in our picture that we have, our DNA is already unwound, but we do still have to unzip it. So this right here is where we're going to start. Let's put unzip right in the middle. We're just pulling those base pairs apart. And we're going to do that with this special guy right here. This is an enzyme. And his name is RNA polymerase. RNA polymerase, ACE for enzyme. All right, and then we're going to draw in, I'll use green. I'm going to change my pen to a little bigger. We're going to draw in right here. I'm just going to color this guy right here. Another enzyme that's going to help us unzip our DNA. And its name is helicase. Okay, so helicase is going to help us unzip, and the RNA polymerase is going to help us build our mRNA. All right, and then our last step that we need to do is close up our DNA, and the mRNA is going to leave our nucleus and go to a ribosome. So right here, this is our mRNA. Our RNA polymerase, this is an enzyme. It's helping to build this mRNA molecule. So let's go ahead and label our picture. This is our mRNA. That's this guy right here. All right, and basically what it's doing is connecting the RNA bases one at a time. Okay, so our RNA polymerase is just bringing in these base pairs and helping them to connect one at a time to build our mRNA strand. Okay, and so once that's completed, we'll need to close it up. So right here, this is closed back up. Our base pairs connect back together. And here is our final mRNA strand. So this is our message, and this guy right here can go to the ribosome. It can leave the nucleus. The DNA can't leave the nucleus, too big. Okay, but this guy's small enough, it can slip out of the nucleus and go find our ribosome so that we can build our protein. All right, and that is the first part, the steps that happen for the first part of protein synthesis, of building a protein. We have to get our instructions from the nucleus to the ribosome with our mRNA.